Today, we are diving deep into one of your body's most amazing survival skills, the power to literally make its own fuel from scratch when the tank is running low. It's a process called gluconeogenesis, and trust me, it's happening inside you way more often than you think. So, let's see how it all works.
So, picture this. You're fasting, or maybe you've just wrapped up a super intense workout. All that easy access sugar in your system, it's pretty much gone. So, what now? Your brain, especially, is a total glucose hog. It can't just shut down and wait. This is the exact reason gluconeogenesis exists. It's the ultimate backup plan. For this explainer, we're going to tackle this in 5 key parts. First, we'll cover the basics of making sugar. Then we'll get into why it's not as simple as just throwing another process in reverse. After that, we'll walk through the clever detours and even an escape plan the body uses to pull this off, all leading up to that big final payoff. All right, let's jump right into our first section, making sugar from scratch. You can really think of this as your body's own personal emergency fuel factory, firing up the moment it realizes the regular supply lines are down. Okay, so the official name is gluconeogenesis. Let's just break that down. Gluco for glucose, neo for new, and genesis for creation. We are literally creating new glucose. Now, you might think of it as just the opposite of glycolysis, the pathway that breaks glucose down. But as we're about to see, it is way more complicated than just hitting the reverse switch. So why is this process so absolutely vital? Well, like we said, it kicks into high gear when you're fasting or after some serious exercise. It's all about making sure critical players, like your brain and your muscles, get the glucose they desperately need to keep running. And where does all this magic happen? The main stage is the liver, though your kidneys and intestines help out a little too. And check out the starting ingredients. This is where it gets really wild. We're not starting with other sugars. The body is an incredible recycler. Using stuff like lactate, that's stuff that builds up in tired muscles, glycerol from your fat reserves, and even bits of protein. It is just metabolic genius. And that leads us to a really important question. If glycolysis turns glucose into pyruvate, why can't we just run that whole pathway backwards? I mean, it seems totally logical, right? But the answer is a hard no. That particular metabolic highway has some major, unmovable roadblocks. And this slide shows the problem perfectly. Glycolysis, the breakdown process, is like rolling a ball downhill. It releases energy. But along that path, there are 3 specific steps that are like massive waterfalls. They release so much energy that they are effectively one-way streets. To go back uphill to build glucose, you can't just push the ball back up the waterfall. You have to find a totally different route around them, a bypass. So the entire challenge of gluconeogenesis really boils down to this number, 3. 3 irreversible steps in glycolysis that we absolutely have to get around. And how the cell does this, well, that's where things get really, really clever. This brings us to the first and honestly the most complicated of these detours. The mission is to turn pyruvate, the end product of glycolysis, back into an earlier molecule. But we can't do it in one shot. The first step on our new route is to turn pyruvate into something else entirely, a molecule called oxaloacetate. And here's that first reaction. You can see we're combining pyruvate with bicarbonate. But I want you to notice 2 super important details here. First, it costs energy in the form of ATP. And second, look at the location. This all goes down inside the mitochondrion, the cell's powerhouse. And that location detail is going to be a huge deal in just a minute. The enzyme that pulls this off, pyruvate carboxylase, is just fascinating. It's less like a simple chemical reaction and more like a tiny molecular assembly line. What it does is it uses energy to grab a CO2 molecule and stick it onto this long, flexible arm made of the vitamin biotin. Then, as you can see in these steps, that arm physically swings the CO2 over to the pyruvate molecule, and bam, we get our new molecule, oxaloacetate. It's an incredible piece of microscopic machinery. Okay, so we've navigated the first big detour. We've made our oxaloacetate. Problem solved, right? Well, not quite. Now we have a brand new problem. You see, the rest of the gluconeogenesis pathway happens out in the main part of the cell, the cytoplasm. But our oxaloacetate, it's trapped inside the mitochondrion. It needs an escape plan. And this is the problem in a nutshell. There is simply no door, no transporter protein that will let oxaloacetate pass through the mitochondrial wall. It's stuck. So how on earth does the cell solve this logistical puzzle? It uses a disguise. Seriously, Inside the mitochondrion, the cell temporarily converts oxaloacetate into a different molecule called malate. Now here's the trick. Malate does have a passport. It has its own special transporter, so it can just waltz right out into the cytoplasm. And the second it's outside, boom, it gets converted right back into oxaloacetate. The whole thing is called the malate shuttle.
But here's the truly genius part, the hidden bonus of this whole escape plan. Look at what's used in these conversions. To make malate on the inside, the cell uses a molecule of NADH. But when it turns back into oxaloacetate on the outside, it makes a molecule of NADH. So this shuttle isn't just moving a molecule, it's actively pumping purchasing power in the form of NADH out into the cytoplasm, exactly where it's going to be needed for the next steps of making glucose.
And that brings us to our final section, the big picture payoff. We have successfully navigated the first major roadblock and solved a tricky transport problem. Now, finally, we can get back to the main event, actually building a glucose molecule.
So back out in the cytoplasm, our freshly escaped oxaloacetate gets immediately converted into something called phosphoenol pyruvate, or PEP. This costs another little burst of energy, from GTP this time. And just like that, we've officially bypassed that first irreversible step from glycolysis. PEP can now travel backwards through all the reversible steps until it hits the next roadblock. This quote really just hammers home how important that malate shuttle is. It's not some weird little side quest. It is a critical part of the whole operation. It's all about balancing the books, making sure the right molecules and the right amount of energy are in the right place at the right time. Without that shuttle, the entire assembly line would just grind to a halt.
And one last, crucial bit of accounting. Remember, glucose is a 6-carbon sugar, but our starting block, pyruvate, only has 3 carbons. So you do the math. For every single molecule of glucose your liver makes, this entire, elegant, complex process we've just described has to happen twice. It takes 2 pyruvates to build 1 glucose.
So there you have it. From a simple starting molecule, through an incredibly complex detour and a brilliant mitochondrial escape plan, the cell builds a brand new molecule of glucose just to keep your brain running. It really makes you wonder what other hidden metabolic detours and ingenious escape plans are working away right this second just to keep you going.